Hello, my name is Elena Hicks, and my presentation is on how thiamethoxam impairs honeybee flight ability. To start, did you know that 1/3 of the food we eat is due to the result of bee pollination? In fact, it is estimated that 1 out of every 3 or 4 bites of the food we eat is due to the responsibility of bees. This has led to the outcome of $15 billion worth of agricultural crops in the United States each year, according to the Natural Resource Defense Council. More specifically, the honeybee Apis mellifera has had an observable overall global decline, which has raised concerns for ecology, crop production, food security, and human welfare. Now everyone is asking the question, what is to blame for this decline? Currently, there is not one explanation. It is likely due to a combination of factors, including disease, invaders such as the Varroa destructor, or pesticides such as neonicotinoids. Neonicotinoids are systemic, meaning they are found in all parts of a plant, including their leaves, roots, stem, nectar, and pollen. This means that bees are not only ingesting neonicotinoids, but they are also acquiring residues on their body that they are then bringing back to the hive. Recent studies have shown that after ingestion of neonicotinoids, it has effects on bee foraging, homing locomotion, navigation, and colony health. This study focuses on the neonicotinoid thiamethoxam. A recent study conducted by Michael Henry, who is a member of the French National Institute for Agricultural Research in Paris, has shown that after ingestion of thiamethoxam, it has effects on reduced forager return rates, which then affects a colony as a whole. This has then led to the next question: does thiamethoxam impair honeybee flight, navigation, flight ability, or both? Our experiment was conducted between September 2012 and September 2014 at the University of San Diego in the Division of Biological Sciences, where we used 19 healthy honeybee colonies. All of our doses were less than worst case scenario, had no effect on increased mortality rates as compared to control, and were all determined based on the rules and regulations set by the European Food Safety Authority. We did 2 treatments, an acute and chronic. In our acute treatment, flight ability was tested before and after treatment. Our control group received a single dose of 10 microliter 2 molar glucose solution. Our treatment group received the same glucose solution, but with 1.34 nanograms of thiamethoxam. Some may criticize that 1.34 nanograms is high or not realistic. However, it is 3.7 times less than the LD50 of thiamethoxam, meaning that it is nearly 4 times less than what it would take to kill 50% of bees after ingestion of thiamethoxam. After the single dose of control or treatment groups, each bee was placed into an individual cage with no food or water, incubated at 30 degrees Celsius for 40 minutes. After 40 minutes, 37 bees from 9 colonies' flight ability was tested using this apparatus. As you can see here, a bee is tethered to one end, a counterweight at the other, and a sensor here. The sensor was able to measure duration, distance, and velocity of the bee. In our chronic treatment, flight ability was only tested after because they were able to feed on the solution for multiple days. Our control group received a 1.8 molar sucrose solution with 0 parts per billion thiamethoxam. Our treatment group received the same sucrose solution, but with either 32.5 or 45 parts per billion thiamethoxam. Both groups were allowed to feed on the sucrose solution for 1 or 2 days. In order to calculate sucrose or thiamethoxam consumption, we used the weight of the syringe and measured the difference in mass loss. 10 empty cages with 10 full syringes were used in order to calculate the average sucrose mass loss based on evaporation. 213 bees from 19 colonies' flight ability was tested using the same apparatus as the acute treatment. And as a result, in our acute treatment, our control group had very similar analogous times for flight duration. However, after treatment, it increased by 78% compared to control. For flight distance, our control group had comparable lengths, but our flight distance had an increase of 72%. In our acute treatment, there were no significant differences on mean velocity or maximum velocity as compared to control groups. In our chronic treatment, flight duration was reduced by 20% at a concentration of 1.95 to 2.9 nanograms per B per day. Our flight distance was also reduced by similar amounts, 23%, at the same concentration, 1.95 to 2.9, as having the largest difference. Mean velocity, unlike the acute treatment, was reduced by 0.15 kilometers per hour and was seen at concentrations of 2.9 to 3.71, which is different than the last two. And our maximum velocity was reduced by the same amount, 0.15 kilometers per hour, at similar concentrations of the distance and duration, 1.95 to 2.9 nanograms of thiamethoxam per B per day. The 1 or 2 days had the same negative effect, so the days of exposure had no significant effects on the amount of bees that did not fly. This is the first time that an experiment showed that acute and chronic sublethal exposures of thiamethoxam had effects on all three of these characteristics: duration, distance, and velocity. Our results showed that in the short term, there was an excitatory effect, but in the long term, there was a depressive effect. This excitatory effect in the short term is likely not beneficial because it reduces the likelihood that bees will be able to come home based on their increased disorientation because they are flying erratically and faster and longer. The inability for bees to be able to return home have been proven to be detrimental based on 2 case studies conducted by Michael Henry. Long-term exposure of thiamethoxam is possible because bees have the choice to feed on whatever source they like. In this case, bees seem to prefer to feed on thiamethoxam treated sucrose as opposed to pure sucrose. This observation has also been observed by other researchers such as Kessler. In the long term, the largest detrimental effects were seen between the concentrations of 1.95 to 2.9 nanograms per B, except for the mean velocity, which was seen at 2.9 to 3.71 nanograms per B per day. Since 1 or 2 days showed the same negative effects on thiamethoxam, it can be concluded that 1 day is enough to weaken flight ability in bees. Thank you.